Good evening, students. So, today I will discuss seed health testing. So, just like human health, seed health is also important. If any seed borne disease reported, so maybe due to seed borne disease in later on, finally, whole plant will get killed. So, that's why we are also going for the seed health testing. So, what is the seed health testing? So, seed health testing is a science of determining the presence or absence of disease causing agents like fungi, bacteria, viruses. So, just I told you, human can be infected with fungi, bacteria, viruses, just like plant also infected with fungi, bacteria, viruses. The animal pests like eelworms, insect in the seed samples. So we don't know, even we are not able to see, but there are many seeds which is infected by the seed-borne disease. So it will express in the later part of their life cycle, or when plant will germinate, then they will show the symptoms of the fungal or bacterial or viral pathogens. So how we are going to detect the seed health testing for the fungi? So simple, by just using by the visual examination with or without a stereo microscope. So this is very simple. Take the sample and just see under the microscope. And infected in dry state for the presence of impurities such as ergot or other sclerosia. So there are different many diseases like in wheat. In case of kernel burnt is very important disease. So that is also, you know what happens. So black powdery mass will get in the seeds. In case of burnt of rice, again seeds are opened with the help of blade and you see whole grains transform into black powdery mass. So from the outside, from the morphological part, you are not going to see, but when you open it, you will see black powdery mass. So what is the alternate procedure? So in that case, you have to treat the seeds, you have to soak the seeds in 0.2% of the sodium hydroxide for 24 hours at 18 to 25 degrees centigrade. And swollen seeds are virtually or visually examined for the shiny jet black colors. I was telling you just, many fungal disease having the what called mycelium for infection, and based on that you can uh, test it, and finally you have to classify. Then next is UV examination. So UV examination, so toxins in fungi gives fluorescent appearance. So if you see the slides, petri dishes, so they are showing the shiny appearance. So by doing so, you can see, you can confirm that presence of fungi is found in the that particular uh, seed. Now next is blotter method. So seeds are placed on the moisture blotter and filter papers at least 20 apart. So you can see here. So all the seeds are sold in the petri dishes, and they are all seeds are at least 20 apart. And blotters are placed in the closed container and incubated for certain number of days, and later examined for the pathogen. So if you leave any seed, so after that you will see if any seed-borne fungus is there or pathogens are there, they will show the symptoms. Now next is agar plate method. So after pre-treatment, seeds are spaced on the surface of 2% malt extract style agar, and after plating, the seeds are incubated at 20 to 25 degrees centigrade for 5 to 8 days. When you keep 5 to 8 days, you will see the also the you will appear to you will see some what called pathogens attack. Now wash method. So seeds are immersed in water with a wetting agents and shaked vigorously to remove fungal spores and hyphae. And excess liquid is removed and material is examined. It is called hyper infection, and based on that you can uh, test it, and finally you have to classify. And this is the PCR method. So already we have discussed in my earlier YouTube what is the PCR. So detailed procedure you can go through the PCR method. So this is the tool by which if you know the primer of certain pathogens, so you can directly take the pathogens, go for the DNA isolation, and just go for PCR. just the set the PCR. If you will get the band of desired size, then we will confirm that particular pathogen is there in the seeds. Now methods to detect seedborne. So bacterial pathogens. So various methods have been developed, and some of the methods are simple, and some of that specialized, namely like serological methods. So one method is called growing out test. So growing out test is the bioassay of working seed sample that involves the sowing of test seeds into the seedlings. Try to focus the point. Sowing of test seed into the seedlings under the condition optimal for the disease development in the glass house or closed environment chambers, either polyhouse. And grow out test have been successfully used for the large number of xanthomonas and pseudomonas. It is called wrong misspelling. Pseudomonas. So if you grow it, so after certain period you will see there is what called appearance of certain fungal diseases. Then indicator test. So working seed sample is sterilized with 2.6% sodium hypochlorite for 15 minutes and enriched with the sterilized water. So this is the what you call. So you have to treat the seed or sterilize the seed with the 2.6% sodium hypochlorite or calcium hypochlorite, and again you have to rinse with the or you have to clean with the sterile water. So seed sample is incubated for 18 to 24 hours in the sterile water, and water suspension is inoculated by the infiltration into the primary leaf node of 10-day-old bean seedling. So you will find appearance of lesions followed by systemic necrosis is positive reaction. Now next is blotter method. So seeds are placed on the moisture blotter and filter papers at least 20 apart. So you can see here. So all the seeds are soaked in the petri dishes, and all seeds are at least 20 apart. And blotters are placed in the closed container and incubated for certain number of days and later examined for the pathogen. So if you leave any seed, so after that you will see if any seedborne fungus is there or pathogens are there, they will show the symptoms. And this is the PCR method. So already we have discussed in my earlier YouTube what is the PCR. So detailed procedure you can go through the PCR method. So this is the tool by which if you know the primer of certain pathogens, so you can directly take the. So again PCR method. So I already told you if you know the sequence of primer, so go for the PCR and you will get the positive. Then methods to detect seed-borne plant viruses. So you know plant viruses are again very dangerous because you cannot control the plant viruses. Just you can control the vectors. So method called biological method. So by growing seeds, so seeds are examined and the abnormal seeds are separated. So this is the very clever manner. Means in the earlier only just examine the seeds and remove the abnormal seeds. Then both normal looking and abnormal looking seeds are grown separately, and seedlings are observed for the characteristic symptoms for the virus disease. So if you grow separately, you can easily distinguish which is the seeds which is infected with the viral disease. And plants are kept free of insect vectors such as mites and whiteflies. So just I was telling ki you have to free from any kind of vectors, so that because vectors are the carrier of all virus, they will go from one plant to other plants and they will spread the virus particles. Then direct seed test. So seeds are examined and abnormal seeds are separated and handled separately. Seeds are soaked in an aqueous medium and then titrated. The slurry produced are then applied to the indicator or test plant, and the slurry containing virus produce symptoms typical for a given virus on the indicator plant. Then serological test. So first is double diffusion test. So this is the seeds to be tested are soaked in tap water, and seed or part that is called embryo of it is titrated, and titrate is transferred to well cut in a diffusion medium like agar gel. An anti-serum specific for suspect virus in the seed is placed in the separate well. So you can see here. So in the time virus particles that is called antigen and antibody molecules diffuse towards one another, seeds combination. That's why it is called double diffusion. Now come to the next method, that is called a radial diffusion test. So procedure consists of charging the wells with the seed or sealing preparation being tested for the presence of virus. When virus molecules are present, they diffuse radially from the well surface and they complex with antibody molecules and precipitate in this region. So again, this is the just interaction between the again antigen and antibody, and finally it will precipitate. So we will observe. So you can see heading of halo result around the charge well. You can see, so this is the interaction. And this is the PCR method. So already we have discussed in my earlier YouTube what is the PCR. So detailed procedure you can go through the PCR method. So this is the tool by which if you know the primer of certain pathogens, so you can directly take the pathogens, go for the DNA isolation, and just go for PCR. Just set the PCR. If you will get the band of desired size, then we will confirm that particular pathogen is there in the sequence. Then next is ELISA, that is called enzyme linked immunosorbent assay. So in this procedure, again I was telling you again antigen antibody rejection. So anti-serum specific for given virus is used to coat the polystyrene plate. So this is the polystyrene plate, and antibody molecules become absorbed. Then seed sample is added to the plate, and it is followed by again adding enzyme that is called labeled with a specific antibody to the plate. So when you use secondary antibody with substrates, it will give color. And by so alkaline phosphate is conjugated to the antibody molecules specific for the virus under examination, and finally enzyme substrate is added to the plate, just I was telling, and hydrolyzed substrate is determined by the measuring the extinction spectrophotometrically or by visual observation. So you can both, because it will give certain color. So based on that color, you can confirm that this particular seed is infected with virus, because you are going to interact with the antibody. Otherwise, you can also measure the extinction coefficient by spectrophotometer. Now, next is serologically specific electron microscope. That is called SSEM. So, ground seeds or homogenized seeds preparation is applied to the electron microscope grid containing an antiserum specific for virus in the question. So, whatever virus you want to, you have to homogenize the seeds and again applied under the electron microscope. Samples from infected seeds lot reveal the presence of virus particle characteristic for the suspect virus. So you can see heading of halo result around the charge well. You can see, so this is the interaction. And after centrifugation, supernatant is discarded and radioactivity of rat is measured, and periods showing radioactivity are counted as reinfected. And again by using again same by PCR or RT-PCR, because nowadays you know coronavirus we are going to detect with RT-PCR. So this is also again virus. And detection of insect and nematodes. So again you can examine the nematodes and insect by stereoscopic microscope, and result is mentioned as the number of insects per weight of the sample. So these are the different detection of the insect and nematodes. So this is the, now come to the detection of external insect injury. So working sample is examined under the magnification that is called 10x or stereoscopic microscope, and absence of insect does not however guarantee that seed lot is free from insect infestation. That is internal injury to seed or internal infestation. Now come to the next method, that is called a radial diffusion test. So procedure consists of charging the wells with the seed or sealing preparation being tested for the presence of virus. When virus molecules are present, they diffuse radially from the well surface and they complex with antibody molecules and precipitate in this region. So again this is the just interaction between the again antigen and antibody, and finally it will precipitate. So we will observe. I was telling is just many fungal disease having the what called mycelium You can see how gauze looks like on the wheat. Again, I'm going to criticize. So these are the nematodes infestation on the wheat. Now you can see root knot nematodes of sweet potato. It is called Mesogyne incognita. You can see there is small knots are visible on the sweet potato. An entire cemetery sample is examined for the visible symptoms of nematode infestation. So these are the gauze of sweet potato. And why? So alkaline phosphate is conjugated to the antibody molecules specific for the virus under examination. And finally, enzyme substrate is added to the plate, just I was telling. And hydrolyzed substrate is determined by the measuring the extinction. So thank you.